Differences of RNA and DNA. There are two differences that distinguish DNA from RNA. RNA contains the sugar ribose, while DNA contains the slightly different sugar deoxyribose, a type of ribose that lacks one oxygen atom. And RNA has the nucleobase or a cell, while DNA contains thymine. So, these are the types of RNA and DNA. We have base pairs, adenine, thymine, guanine, cytosine, and sugar phosphate backbone. Base pair. Base pair is a fundamental unit of double-stranded nucleic acid consisting of two nucleobases bound to each other by hydrogen bonds. Adenine. Adenine is one of the four nucleotide bases in DNA, with the other three being cytosine, guanine, and thymine within a double-stranded DNA. Thymine is one of the four nucleotide bases in DNA, with the other three being adenine, cytosine, and guanine. Guanine is one of the four main nucleobases found in the nucleic acids DNA and RNA, the others being adenine, cytosine, and thymine. Cytosine is one of the four nucleobases found in DNA and the RNA, along with adenine, guanine, and thymine. And sugar phosphate backbone forms the structural framework of nucleic acid, including DNA and RNA. This backbone is composed of alternating sugar and phosphate groups and defines directionality of the molecule. Structural differences of DNA and RNA. This is the example of DNA and RNA of genetic information. Deoxyribonalic acid and DNA and ribonucleic acid RNA are perhaps the most important molecules in cell biology, responsible for the storage and reading of genetic information that underpins all life. They are both linear polymers consisting of sugars, phosphates, and bases, but there are some key differences which separated the two. Pinterest DNA reflection. Summary of DNA reflection. This is the example of DNA reflection, and DNA contains a sugar deoxyribose, while RNA contains the sugar ribose. Is a double-stranded molecule, while RNA is a single-stranded molecule. Stable under alkaline conditions, while RNA is not stable. DNA and RNA perform different functions in humans.
Transcription. Transcription is the synthesis of RNA from a DNA template where the code in the DNA is converted into complementary RNA code. Transcription in the synthesis of a protein from a messenger RNA template where the code in the messenger RNA is converted into an amino acid sequence. Transcription is a process of DNA. The segments of DNA transcribe into RNA molecules that can encode proteins said that to produce messenger RNA. Other segments of DNA are copied into RNA molecules called coding RNA. So, transcription is uses the genes as templates to produce several functional forms of RNA. Next is transcription. Transcription annotation occur when RNA polymerase protein binds to the promoter in DNA and forms a transcription annotation complex. Promoter directs the exact location of the annotation of transcription. So, annotation transcription annotates with RNA polymerase binding to the promoter region in the DNA. The transcription of factors and RNA polymerase binding to the promoter forms a transcription annotation complex. Next is termination transcription. RNA transcript is released and polymerase detaches from DNA. DNA rewind itself into a double helix and as unaltered throughout this process. Elongation. Elongation. RNA polymerase in the 5 to 3 direction. So, elongation during the transcription, the RNA polymerase after the initial abortive attempts traverses the template strand of DNA in 3 to 5 direction, producing a complementary RNA strand in 5 to 3 direction. As the RNA polymerase advances, the DNA strand that has been transcribed remains to form a double helix. Next is antibiotics is inhibited by rifampicin and 8-hydroxycycline. So rifampicin is rifampicin is an antibacteria while the hydroxycycline is an antifungal. Next is a simplified diagram of RNA synthesis and processing. So, both DNA and RNA are nucleic acid, which use base pairs of nucleotides as a complementary language. So, during transcription, a DNA sequence is read by an RNA polymerase, which produces a complementary anti-parallel RNA strand called a primary transcript. So, here are the steps of transcription. Number 1, RNA polymerase, together with one or more general transcription factors, binds to promoter DNA. 2, RNA polymerase generates a transcription bubble, which separate the two strands of the DNA helix. This is done by hydrogen bonds between complementary DNA nucleotides. 3, RNA polymerase adds RNA nucleotides, which are complementary to the nucleotides of one DNA strand. And 4, RNA sugar phosphate backbone forms with assistance from RNA polymerase to form an RNA strand. 5, hydrogen bonds of the RNA DNA helix break, freeing the newly synthesized RNA strand. 6 is, if the cell has a nucleus, the RNA may be further processed. This may include polyadenylation, capping, and splicing. And last, the RNA may remain in the nucleus or exit to the cytoplasm through the nuclear pore complex. So if the stretch of DNA is transcribed into an RNA molecule that encodes a protein, the RNA is termed messenger RNA. The messenger RNA in turn serves as a template for the protein synthesis through translation. Other stretches of DNA may be transcribed into small non-coding RNA such as microRNA, transfer RNA, small nucleolar RNA, small nuclear RNA such as ribosomal, and a long non-coding RNA. So in overall, the RNA helps synthesize, regulate, and process proteins. It therefore plays a fundamental role in performing functions within cell. So i- the purpose of transcription is to make the RNA copies of individual genes that the cell can use in the biochemistry. Next is there are two types of gene interaction: the intragenic interaction and the intergenic interaction. So intragenic interaction, two alleles of a gene which are present on the same gene locus on the two homologous chromosomes react to produce modified phenotypes. So intergenic interaction is also called our interallelic interaction. So this type of interaction that involves two alleles of a gene present on the same locus of the two homologous chromosomes. So there are types of intragenic: incomplete dominance, multiple alleles, codominance, and lethal genes. So incomplete dominance, multiple alleles, and codominance is the example of intragenic. So incomplete dominance is a codomin is a new phenotype is produced when both the alleles are unable to completely dominate and rather produce a mixed phenotype. Codominance is when both the alleles dominate equally and express both their phenotype at the same without getting mixed up. Multiple alleles is when there are more than two alleles for a gene in the population. The phenotype depends upon which two alleles exist in a diploid individual. So codominance is a coat color in short home cattle, and lethal genes is a body color in the mouse. Next is intergenic interaction. So interaction between genes on different chromosomes producing changed phenotype. So this type of interactions occur between the alleles of different genes present on the same or different chromosomes. So includes epistatic genes, duplicate genes, complementary genes, and supplementary genes. So epistatic genes is a phenomenon in genetics in which the effect of the gene mutation is dependent on the presence or absence of mutations in or more other genes, respectively termed modifier genes. In other words, the effect of the mutation is dependent on the genetic background in which it occurs. Next is duplicate genes. So gene duplicate, it can be defined as any duplication of a region of DNA that contains a gene. Complementary genes. Complementary genes are genes that mix together to produce a new distinct phenotype. And last is supplementary genes. So supplementary genes are genes that both contribute to a single characteristic where one gene can mask the effect of the other.